Hello friends, today we are going to talk about the nervous tissues under the nervous system. So let's begin. There are main two types of nervous tissues, which are neurons and neuroglia. Basically, neurons are the working units of nervous system and neuroglia are the supportive cells to the neurons. Let's talk about the neurons. Neurons are the working units of nervous system that generate and transmit nerve impulses. As well as we can call this as the basic element of the nervous system. Around 10 to power 11 neurons in the human body. These are excitable cells. Each neuron consists of cell body and its processes such as axon and dendrites. Neurons are very considerable in size and shape, some being the largest cell of the human body. Neurons cannot replicate. For survival, they need a continuous supply of oxygen and glucose. Neurons can normally synthesize chemical energy, which means ATP, only from glucose. Also, these neurons generate and transmit nerve impulses called action potential. The initial strength of the action potential is maintained throughout the length of the neuron. Transmission of nerve signals is both electrical and chemical. But the action potential traveling down the nerve axon is an electrical signal. But nerves do not come into direct contact with each other. Because of that, signals between a nerve cell and neck cell is nearly always chemical. Let's move on to the cell body of the neurons. Like another cells, this cell is too small to be seen with the naked eye. And neuron cell body form the gray matter of nervous system found at the periphery of the brain and center of the spinal cord. In CNS, group of cell bodies are called nuclei. In PNS, it called ganglia. But basal ganglia situated within the cerebrum, they are dark in appearance. Let's move on to the neurites. Neurites are the extensions of cell bodies form the white matter of nervous system. Earlier, we called that cell bodies form the gray matter of nervous system and neurites are formed the white matters of nervous system. There are mainly two types of neurites, which are axons and dendrites. Okay, let's move on to the axon. Each nerve cell has only one axon. This begins at a tapered area of the cell body, the axon hillock. You can see there are so many tapered areas in this nerve cell like these. These are tapered areas. Axon hillocks also begin at a tapered area of cell body like this. This tapered area is called axon hillocks. So, these axons carry impulses away from the cell body to this direction, and axons found in the brain in group called tracts and in periphery of the spinal cord. Important point is these axons found deep in the brain and found periphery of the spinal cord. So outside the brain and spinal cord, axons are referred to as nerve or nerve fibers. Usually, these axons are much longer than the dendrites. You can see that axons are much longer than the dendrites. Membranes of the axon is called the axonema. Large
axons and those of peripheral nerves are surrounded by a myelin set. This myelin set consists of a series of Schwann cells. In this picture also you can see the series of Schwann cells. These are the Schwann cells. So each one is wrapped around the axon, so that is covered by a number of concentric layers of Schwann cells plasma membrane. As you can see, there are a number of concentric layers of plasma membrane of the Schwann cells. Between the layers of plasma membrane is a small amount of fatty substance called myelin. That's why we call this the myelin set. To this we call myelin set. The outermost layer of the Schwann cell plasma membrane is the neurolemma. Hope you remember that we talked earlier. The membrane of the axon is called the axolemma, as well as the membrane of the Schwann cell plasma membrane is the neurolemma. So these are important, axolemma and neurolemma. There are tiny areas of exposed axolemma between adjacent Schwann cells called knots of Ranvier. These are the knots of Ranvier. In here, like in these places, there are small spaces. We call this knots of Ranvier. You can see clear picture from here. These are the knots of Ranvier. Like these places, we call knots of Ranvier. This assists the rapid communication of action potential in myelinated neurons. Also, this myelin set acts as insulation and it helps to speed up the passage of impulses along the axon. So let's move on to the unmyelinated neurons. Postganglionic fibers and some small fibers in CNS are unmyelinated. In this, several axons are embedded in one Schwann cell. Like this, if you think these are the axons, number of axons are embedded in one Schwann cell like this. These are the concentric layers and these are the axons. So in unmyelinated neuron, several axons are embedded in one Schwann cell. There is no exposed axolemma in this unmyelinated neuron. Conduction of action potential is significantly slower in unmyelinated fibers than the myelinated fibers. Let's talk about the dendrites. We told that nerve cell has one cell body and its processes such as axons and dendrites. So we talk about the axons. Now we are going to talk about the dendrites. Dendrites are the short processes that receive and carry incoming impulses towards the cell body. Same structure as axons, but are usually short and branching. In motor neurons, dendrites form part of synapses. In sensory neurons, dendrites form the sensory receptors. Classification of neurons. We can classify neurons based on the function and based on the structure. Based on the function, we can classify neurons into main three parts, which are sensory neurons, interneurons, and motor neurons. Sensory neurons allow to receive information from the outside the world through sensors. Interneurons connect and communicate neurons within specific region of the CNS. As you can see in this picture, interneurons in here connect and communicate neurons within the specific region of the CNS. Motor neurons transmit signals from the CNS to the effector organ. So let's move on to the classification of neurons based on structure. Based on structure, we can classify neurons into main three parts, which are multipolar neurons, bipolar neurons, and unipolar neurons. Multipolar neurons, cell types have a single axon extending from one end of the cell body and dendrites branching from other side of the cell body. Most times, brain and spinal cord. Most times, brain and spinal cord. This is the neurons that we have normally seen. The next one is bipolar neuron. Bipolar neuron only associated with apparent impulses. They have a single axon projecting from one end of the cell body and long dendritic tree extending from the other end. We can see in this type of neuron in retina of the eye. Third one is unipolar neuron. Unipolar neuron has one single process. This process divides into two branches close to the cell body, and one branch travels from the cell body to the spinal cord, while other travels from the periphery to the cell body. Okay, let's talk about the neuroglia. Neuroglia are non-excitable glial cells. There are around 1 trillion glial cells in the body. Neuroglia act as supporters to the neuron. Glial cells continue to replicate throughout life. There are 4 types of glial cells supported to the neurons in the CNS. These are astrocytes, oligodendrocytes, ependymal cells, and microglia.
Astrocytes. Astrocytes are the most abundant neuroglia and they are star-shaped. Astrocytes have fine branching processes and lie in a mucopolysaccharide ground substance. These are the fine branching processes of these astrocytes. At the free ends of some of the processes, there are small swellings called foot processes. There are small swellings. These small swellings we call foot processes. Second cell type is oligodendrocytes. Oligodendrocytes are smaller than astrocytes. Like Schwann cells in the PNS, oligodendrocytes form and maintain myelin in the CNS. So, main function of the oligodendrocytes is form and maintain myelin in the CNS. And oligodendrocytes are found in clusters around nerve cell bodies in grey matter and are located adjacent to and along the length of myelinated nerve fiber. You can see here this is the oligodendrocytes and it's located adjacent to and along the length of the myelinated nerve fiber. So next we are going to talk about the ependymal cells. Ependymal cells form the epithelial lining of the ventricles of the brain and the central canal of the spinal cord. It forms epithelial lining of ventricles of the brain and central canal of the spinal cord. Also, this form the choroid plexuses of the ventricles secrete CSF. CSF means cerebrospinal fluid. Someone also have cilia that assist the flow of CSF. You can see here, this is the cilia. This assists the flow of CSF. The fourth cell type is microglia. This is the smallest and least numerous glial cells. Okay, we talked about neuroglia in the CNS. Now we are going to talk about the neuroglia in the PNS. Neuroglia of the PNS completely surround axons and cell bodies. There are two types of glial cells in the PNS, which are Schwann cells and satellite cells. Firstly, we are going to talk about the Schwann cells. Schwann cells are similar in function to oligodendrocytes and microglial cells, providing myelination the axons in the PNS. So one of main function is of the Schwann cells is providing myelination the axons in the PNS. They also have phagocytic activity and clear cellular debris that allows for regrowth of PNS neurons. So this is also a function allows for regrowth of PNS neurons. So the next cell type is satellite cells. Satellite cells are similar in function to astrocytes. Small cells that surround neuron in sensory, sympathetic and parasympathetic ganglia, helping to regulate the external chemical environment. So in here, this is a Schwann cell and this is a satellite cell. So both of these cells are supportive cells to the neurons. Okay friends, in this video I talked about the nervous tissues. On my next videos, I'm going to talk about more of the nervous system. So till then,